Hi, and welcome to this video where we're going to be running through the AS level biology paper, Breadth in Biology from 2016. So first thing I recommend is to follow the link underneath the video in the description to get this paper and to do it. And then you can use this paper to kind of see where you went wrong and to kind of understand the thought process for this paper. Okay, so if you need to do that first, go ahead and do that. Alternatively, if you kind of want to get a bit of a helping hand with this paper, you can kind of listen to the intro of each question, and then you can pause the video at the appropriate time and try and complete it, and then see how you do. So you get a bit more immediate feedback. Up to you. All right, so we're going to start with the multiple choice. And what I'm going to do actually is just going to put the mark scheme up here first. So here it is. Okay, so here's the mark scheme for the multiple choice section. So first of all, go ahead and mark all the multiple choice questions that you've done. And then I'm going to do a run through of each question, how you get the answer, so you can sort of see the questions that you went wrong on how you work it out. Okay, so let's get started now that you've marked the multiple choice and look at the working. Okay, so multiple choice, question one. Actually, this was a tough one. This is something that I struggle sometimes with, remembering all the magnification. Now, light microscope is definitely not 400, that's too low, so I would have probably just, first of all, sort of highlighted these. Okay, it's those are the possibilities. Resolution in nanometers. Well, actually it's kind of difficult to see anything below around about a half a micron. Clearly, but with sort of oil immersion microscopy, you might be able to get a bit lower, but 100 is too low. So then I would sort of highlight these two. Then we've got transmission and scanning. So you need to remember that transmission electron microscope has a higher magnification than a scanning microscope. So which ones work from that? Well, this one doesn't work because that's lower than that. So therefore we are left with this one, 0.2 is the appropriate resolution. 10 is way too large because you can actually see the membrane, which is only about 7 nanometers across. So yeah, it's lower than 7, so 0.2. So C. Okay, so that's the first one there. All right, moving on. Hopefully this next one should be a bit more simple. Carbohydrates such as starch are made from monosaccharides joined together. This is just something you need to remember. So glycosidic bonds join two monosaccharides together. Remember, we can have things like 1,4 glycosidic bonds or 1,6 glycosidic bonds. Those are different types. Question 3. Some inorganic ions have roles in enzyme-controlled reactions. These are the two ions that you need to know about, zinc and chlorine. You just have to remember which way round it is. So for amylase, it's chlorine minus. In fact, you can eliminate some of these because you know that Cl2 minus, that's not a thing, so you can just get rid of that straight away. So it's definitely not C. Chlorine minus is the one for amylase, and zinc 2 plus is for carbonic anhydrase. If you know your chemistry, you might have also recognized that zinc plus is not a thing. Okay, 4. So the answer here is D, sorry. 4. Which of the following statements, A to D, about the nature of the genetic code is incorrect? It is degenerate. That's right. So that means that, for example, you could have two different codons that mean the same thing. These are ones I made up. I don't know them off by heart, but let's say, I don't know, like ACG and ACC, for example. They might mean the same thing. It is a triplet code, 3 letters. It is not overlapping. It is universal. So overlapping would mean that if you had a sequence, you would, if, for example, reading this sentence, you would be like WHI and then HIC and then ICH. So there's like, that would be an overlapping reading frame, which it is not. So the answer is C. 5. Ventilation involves parts of the mammalian respiratory system, which describes inhalation. Okay, well, you can, if you were stuck on this one, you can sort of do it yourself. The rib cage is moving up, not down, so we can immediately cross out that one. Nope. We can really cross out that one. And then we've also got the diaphragm pulling down, contracting. So the answer is therefore C. So this is, yeah, upwards. External intercostals contract. Yeah, so the external intercostal muscles are on the outside of your ribcage that pull your ribcage up. Internal intercostals pull your ribcage down. So the answer is C. 6. Which of the following structures, A to D, are found in prokaryotes and in eukaryotes? Not this, okay? Peptidoglycan is only found in prokaryotes. Let's just double check that these circular genomic DNA, I wouldn't. Oh, and yeah, sorry, yeah, no, I got confused. I was confused about what the question said. Yeah, so that's not found because that's only in prokaryotes. Nucleus surrounded by nuclear membrane. No, that's only in eukaryotes. Ribosomes, yes, ribosomes are found in both. So that's prokaryotes, and that is eukaryotes. So D, prokaryotes and eukaryotes. 7. The pressure changes in one mammalian cardiac cycle are shown in the graph. When is ventricular systole? So this is when the ventricle is contracting and pressure is rising. Okay, so it starts right about here when this starts to go up, and then it's actually probably going to stop right about here once the pressure has finished rising. So that's really about 0.2 to 0.3, which is option B. Just so you are aware, this little bump here would be atrial systole, and this phase would be the yellow phase here is ventricular systole there, and then everything else would be the diastole. This would be the diastole. Okay, on to 8. All right, I like this one. This is kind of one of these how science works questions. So for these questions, you always want the way that they're trying to get you to think is they're trying to get you to think cautiously, okay? So you don't want to look at some data and make a conclusion that you can't be sure of, okay? So certain things might seem, oh yeah, that seems like it might be right, but you can't be sure. So which things can we be sure of? So first of all, A, an increase in herd immunity resulted in fewer deaths with measles. Seems reasonable. Can we be sure? No, because this is not talking about causality. It's just an association. So resulted in, no, that's causality. We can't really say that caused one thing caused the other. B, the highest number of measles cases occurred when MCV coverage was at its lowest. Well, when was the highest? Let's find it. Here it is, right there. Was the vaccine at its lowest? Actually, no, it wasn't. It was there. Okay, and the lowest was there, so this is not correct. Almost correct, but not. A 90% MCV coverage resulted in fewer than 500,000 cases each year. Okay, well, when was 90%? It was up here. And in those years, did we have fewer than 500,000? Actually, yes. Okay, so it's from here. From 2006 onwards. So the answer is C. This one, there is a positive correlation between the number of measles cases and MCV coverage. No, it is a negative correlation, okay? Not positive. So the answer is C. Tricky one there. Remember, the motto of this is be cautious in your conclusions, okay? What can you be sure of? Number 9. Okay. Wildflower species, results are below. Which field will have so which of the options is greater? Which of the options is correct? Field G will have a greater Simpson's diversity index. Yeah, it will. Because it has better species richness. Well, no, same species richness, but this has better evenness. Field H has greater evenness. No, it doesn't. Field H will have a greater index. No. Field G has a greater spiritual richness. No, because they have the same. So it's A. Because of the greater evenness in field G. 10. And then moving on to 11. Okay, so 10. Which of the following lists the 3 domains of life? It is archaea, bacteria, and eukaryota. Hang on a minute. Or is it? That's basically right, but let me just check. Bacteria, no, because those are the same. No, no, that's not right because protists are eukaryote. Protist plant. No, okay, yeah, it's definitely A. Sometimes we have multiple ways of expressing this. Sometimes it's Archaebacteria, Eubacteria, and Eukaryota, but just different Latin ways of saying the same concept. But yeah, 10A is correct. Okay, question 11. Should investigate some solutions X, Y, Z using paper chromatography, and here are the results. Which of the following options A to D is the RF value of Z? Okay, so I can't use a ruler on this computer screen, but what you need to do is you need to do this distance from the center of the spot to the line here. So this distance, let's call this the distance, Z. And then this, you need to measure this distance all the way to the solvent front. Let's call that distance SF for solvent front. And the RF value is going to equal Z over SF. Okay, so that is going to come out as 0.63. Okay, you can sort of that looks approximately right, because if that whole distance is 1, it's over halfway, but it's it's probably not 3/4 of the way. So yeah, it's going to be 0.63. It's definitely not going to be that, because this is actually impossible. You cannot have an RF value bigger than 1. So not B, it is A. All right, moving on. 12. Pressure varies in different parts of the mammalian circulatory system. Which of the following options correctly completes the table below? Well, I'm just going to fill it in. Blood and aorta is super high. Tissue fluid is low. Lymph is low. And vena cava is low. So high, low, 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 which is going to be C. 13. A diagram of a potometer is shown below. Which of the following options, A to D, is a precaution that is not needed when setting up a potometer? Well, I'm going to read through all these first. I don't know about this one. It seems, I don't know. I mean, it'll probably help transpiration to remove the excess water from the leaves. So I'm going to come back to that. The screw kit must be opened while taking the readings. No, definitely not that. The chute should be cut whilst underwater. Yes, that is necessary. There should be no extra air bubbles. Yes, that's necessary. Okay, yeah, well then now it seems pretty clear that B. I was a bit unsure about A, but thinking about it, yeah, if there is water on the leaves, it's going to be blocking the stomata, and it's going to affect your results. So yeah, A is true as well. 14. The image shows stages in mitosis. What have we got here? This looks to me to be quite clearly metaphase, because they're all lining up down the middle. So the answer is B. Okay, question 15. Here's an antibody which identifies the region that allows the distance between the antibody sites to vary. Okay, well, we're talking about the hinge region here. So that's going to be, in actual fact, that's going to be C. Because if this is kind of an antibody, we can bind over here at my fingertips, and we can bind over here at my fingertips, but those binding sites can get quite close, or they can flex quite widely because of that hinge, which allows them to accommodate pathogens where the antigens are spaced differently. Okay, all right. 16. A group of students were investigating the diffusion of molecules across membranes using a model cell. Okay, these ones are always quite tricky. All right, so first we really got to analyze this. We've got to figure out what's going on here. All right, so we've got a model cell, which is a bag of viscine tubing. So the viscine tubing is going to mimic a biological membrane. We've got the extracellular fluid, and we've got the cytoplasm. We've got to make sure we look at this here. It contains starch and amylase. So immediately we can sort of, make links between those two. Glucose is going to be produced, isn't it, inside the bag, because we've got a starch and an enzyme. Biochemical tests were used to identify the types of molecules present. The results are shown in the table below. Okay, so this question, actually, first time it threw me slightly, but let's see how you work it out. Well, let's come back to Benedict's test at the end, because I think this is the most tricky. First of all, let's look at the other two. Okay, so burette test. What is that test for? Test for protein. Is there any protein here? You might say there's no protein anywhere. You'd be wrong, because amylase, what is it? It's an enzyme, and an enzyme is a protein. So the only place we're going to have protein is going to be inside the sort of cytoplasm. So we're looking at these three. Oops, no, we're not looking at that one because that says both. So it's either A or D straight away. Now let's look at the iodine test. Iodine is going to be testing for starch. So where is the starch? The starch is on the inside. So yeah, okay, it doesn't really give us much. So all of these could be possible. Actually, not that middle one, because starch is too big to escape the Visking tubing. So we've already eliminated C for sure, and we've also eliminated B. So we're just down to A or D. So now it comes to this one. So where would we find glucose? Would we find it in the extracellular fluid, or would we find it in the cytoplasm? Now, it's a kind of a tricky one. I don't love this question. I think it's slightly misleading. The reason is glucose will be produced inside the cytoplasm. However, then it will move out, it will diffuse out because it can get through the membrane. Okay, so therefore, we're probably going to say that the answer is A, because the glucose will start building up in the extracellular fluid. Although I think that there would still be some glucose present in the cytoplasm as well, because that's where the glucose is produced. But the best answer in this case is going to be A. Okay, question 17. This is a bit of a pain. You've got to do quite a lot of calculations. So first of all, we're going to have to work out the surface area. Then we're going to have to work out the volume, and we're going to have to do the ratio. So the surface area is 6 times the square, okay? So this is 6 times 1, which is equal to 6. This is 6 times 2 by 2, so 6 by 4, which is equal to 24. This is 6 times 16, because 4 by 4 is 16. I've got to do that on my calculator here. That's 96. And this is going to be, so 6 squared is 36 times 6 equals 216. Okay, so that's the surface area up here. Now let's do the volume. Slightly easier, we just have to cube it. So that's just 1. 2 cubed, 2 by 2 by 2 is 8. 4 cubed is 4 times 4 is 16. 16 times 4 is 64. And 6 cubed is 6 times 6 times 6 equals 216. Okay, so therefore, this is going to have a surface SA to V ratio. This is surface area to volume, so this is going to be 6 to 1. This is going to be 3 to 1. Ta-da! There we go, that's what we want. And let's just work the rest of them out. This is going to be like, I can't do that in my head. It's going to be less than 2, is it 1.5 to 1, I think? I think it's 1.5 to 1, and this is 1 to 1. I'm not sure about this. I haven't done it on my calculator, I've just guessed. But we know this is the right answer. There it is, ta-ta-ta. The answer is B. All right. They really make you work for these 1 markers, don't they? Moving on, 18. Okay, diagram of a stained blood smear observed under a light microscope is shown below, which is a neutrophil. Just got to recognize this straight off. It is B. The reason is it's got a lobed nucleus. Okay, you can see that lobed nucleus. That's what you're looking for. This one, if you wanted to know, this is a macrophage or a monocyte, depending on the question. This is a lymphocyte. And this is a red blood cell, or erythrocyte. 19. Which of the following options, A to D, is a primary defense mechanism against pathogens? Nope. Nope. Question mark, question mark. Now, primary defenses means stop things getting in. So the best one is D, blood clotting. The reason I went is because in the syllabus it says that inflammation is primary, which I disagree with, but that's what the syllabus says. I would class inflammation more of a secondary thing because it's when it's in the body, and phagocytosis and inflammation kind of are very closely linked. But phagocytosis, I think, yeah, on reflection, phagocytosis definitely secondary. Blood clotting is the best 1 there for D. 20. Pathogens cause disease are transmitted from individual to individual in a variety of ways. Which of the rows A to D in the table is correct? Okay, well, we'll just go through it. Athlete's foot, yeah, it's a fungus. Direct and indirect contact, yes, that's true. Okay, well, that was easy, it's A. Let's just double check, okay? HIV virus, HIV AIDS, that is a virus. Indirect contact, no. We need sort of bodily fluid transfer. Malaria, no, it's not a bacteria, it's a protoctista, but vector is correct. Tuberculosis, no, that's a bacteria. And direct contact, no, because it's droplet infection. So answer is A. All right, okay, so we've gone through the multiple choice questions. So hope you made some notes on the ones that you did go wrong on. And now we're going to jump into the longer answer questions. If you need to pause the video now, grab a cup of tea, refresh your mind, and then we'll come back in a couple seconds and we'll get going. Okay, on to section B, longer answer questions. Start with question 21. So always a good idea to kind of highlight key information as you go. So we're investigating water potential. This is a little reminder that we need to talk about water potential if we're talking about osmosis. We've got the tube, we've got the sucrose solution here, discs of potato tuber tissue. Okay, discs were placed in the boiling tube containing the sucrose solutions of different concentrations for hours. And then we had the percentage change of mass was then calculated. So we can see here, it's fairly clear that all these are positive. And okay, so the question says, state 2 details of the procedure that must be followed to obtain valid results. Well, there's so many things that you need to do to obtain valid results. Things to do with the discs of potato. So they need to be the same size, the same surface area, from the same type of potato or the same potato potentially. The volume of the sucrose solution is going to have to be measured correctly. I'd probably go with the surface area and the type of potato for my answers there. And I'll quickly flip to the mark scheme so you can see the things that you could say. Okay, so pause the video here, have a look. Here are the different things that you could say. Do kind of notice that, you know, when sometimes things are underlined, like volume of solution, you know, you really have to be precise. You couldn't, for example, say the amount of solution, because that would, you want to say volume or concentration. Amount is a bit vague, for example, okay? So do really make sure you've worded it in exactly the same way. Okay, now let's go back. A part 2, explain how a student could use the data to determine the water potential of the potato tuber cells. We're going to look at the mark scheme in just a minute, but essentially what you do is you plot a graph like this, and along here is percentage change, and along here is the concentration from 0 to 0.5, 0.45 I think it is, and you have the values and you kind of graph them like this. And they're going to look a bit like this, and then you draw a line of best fit like that. Whoops, I mean that's not a good line of best fit, but you see what I'm trying to draw. And it's the point at which the line of best fit crosses over. I've got to get rid of that line of best fit, that's awful. Let's try that again. That's better. And it's the point at which it crosses over here is what you would expect the water potential to be, okay? Now let's look at the mark scheme, and you can mark your answer. Here it is, okay? So we're looking for the idea that there's no change of mass. You want to say that it's between the 0.2 and 0.3 figures. That's where it goes from positive to negative. And then you get marks for saying plot the graph. And you have to be specific here. Plot graph of concentration of sucrose solution against percentage change in mass, and then find which sucrose concentration gives no change in mass of the potato when it crosses the axis. So here, for example, e.g. the point where the line of best fit crosses the x-axis when the y-axis equals 0 is the concentration of sucrose in the potato discs. This will get the mark. This is a good explanation here. I like this. This is good. Okay, if you want to look at that a bit more, pause the video now, and then we'll move on to the next question. Okay, B. Okay, well, here is a potato tuber disc. It's in the high sucrose solution. So it's in the 0.45 moles sucrose solution. What is going on here? Well, let's just label everything, shall we? X to me looks like cell wall. This is cell membrane. And Z is the tonoplast. Tonoplast membrane, and so U being the vacuole. V being cytoplasm. W. What is W? Well, this cell is showing plasmolysis, okay, because it has been placed in a solution of very low water potential, so water has exited the cell into that solution. So kind of water has moved out via osmosis from the cell. So what is taken this space? Well, this space here is actually just the same as the solution that it's been placed in. So W is the sucrose solution, okay? And the mark scheme is quite specific on that. You have to actually say the sucrose solution. You might have said water, but that's not quite right. It's the solution. The external solution would also be acceptable. I'll show you the mark scheme now. There it is. So see, notice how in the mark scheme, you know, we need to have certain things underlined. So would I have even, no, see, look, I messed this up, okay? Bad me. So I needed to say cell surface membrane. I think I just said cell membrane because I was going a bit too quick. Tonoplast would have been accepted here, or vacuole membrane. Yeah, let's correct that. Cell surface membrane. You do need that solution. Really focus on the mark scheme. The underlined words are the most important thing. You've got to hit those underlined points. So cell surface membrane. Cell surface membrane. Okay, moving on down to here. Okay, halophytes. These are things that like salt. So they can live in soils with very low water potential. Salt marsh communities suggest and explain how the root hairs are able to absorb water by osmosis from the soil of the salt marsh. Well, if the root hair is going to absorb water, the root hair cell has to have an even lower water potential. So we're going to need to think along the lines of what features could enable those cells of the halophyte plants to have an even lower water potential. So they're going to have to actively transport in mineral ions at a high rate to generate a very low water potential to allow osmosis to continue. Okay, let's look at the mark scheme now. Here it is. So we have to talk in terms of water potential. We have to say lower, not just low, but lower, lower water potential. Notice how it underlines the ER because it's a comparative statement. It's lower than the outside. So talking about actively transport mineral ions into the root hair cells, and the root hair cells contain a high concentration of mineral ions or salts. Okay, all right, so that's question 21. Pause there if you need to check that mark scheme. And now let's move on to 22. Okay, question 22. Even the smallest DNA molecules are very long. The kilobase is 1,000 base pairs of a DNA molecule. 1 kilobase of double-stranded DNA has a length, so 1 kb, I'll do that in a different color actually, 1 kb has a length of 0.34 micrometers. The DNA in the nucleus of cell from fruit fly is 5.6 centimeters long. Calculate the number of kilobases in the DNA. Okay, maths, fun stuff. Okay, right, so 5.6 centimeters. First of all, we're going to have to convert this into microliters. So 5.6 centimeters equals 56 millimeters equals 56,000 micrometers. Okay. 1 kilobase has a length of 0.34. So we're saying how many kilobases fit into this. Okay, so now we need to do 56,000 divided by 0.34 micrometers. This is going to give us a massive number. Okay, so we're going to find out how many kilobases it is. It's going to be big. So 56,000 divided by 0.34. And that's a lot. Okay, so it's 100 and give you see, look at that. That's so important right here. This is a little tricky little thing here. Give your answer to the nearest whole number. So I guarantee that one of the marks is going to be just for giving that to the nearest whole number. So this is the number I've got on my calculator, 164,705.8. Okay, so what do I round it to? Nearest whole number, basic maths here, 164,706. Okay, am I right? Let's check the mark scheme. Aha, yes I am. So look, we've got to have 2 marks there for rounding it correctly. Okay, so don't forget to round correctly. The DNA of the fruit fly was analysed, and 22% of the bases were adenine. What percent were guanine? Okay, well let's just do a quick remembering that A binds to T and C binds to G. So therefore, 22% equal A. So therefore, 22% is T, because there must be equal amounts. So together, A and T equals 44%, which must mean that C plus G is 100 minus 44. So that's, it's not 66, is it? It's 56. So 56%. And if we divide that by 2 to get either C or G, then we're going to get the answer. So 100, I'm just double checking mine working, 100 minus 44 equals 56, divided by 2 equals 28%. Okay? Mark scheme, yep, correct. All right, moving on. A DNA molecule contains polynucleotide strands. Individual nucleotides are joined together to make a polynucleotide strand. What type of chemical reaction joins things together? This is a sort of basic fact, and it is a condensation reaction. Remember, condensation, joining things together. Hydrolysis splits things apart. Name the chemical released when the bond is formed. What is formed during a condensation reaction? Well, we've got a bond is formed plus water. Oh, this, I bet this is, you've got to write water, okay? This is one of these horrible, horrible things that the OCR board does. I bet, let's look at the mark scheme. Look, because it says name the chemical released. Name it. You have to give it the name, which is water. I bet if you write H2O, it's not going to give you the mark. Ooh, it does give you the mark. Okay, except H2O. Correct formula only. I have seen, though, I've seen it where they don't give you the mark for H2O because that's not the name of it. That's the formula of it. And when I see that, I do a little scream inside because I think it's really, really horrible, but you should be aware of that. Okay. A DNA molecule contains 2 polynucleotide chains. How are they held together? Well, we need to talk about this A to T, C to G. So, the mark's going to be that. So A to T, C to G. We could also say purines to pyrimidines. I put these in the wrong order. So purines are A and G are purines, and C and G are pyrimidines. We talk about the hydrogen bonding between them. We can say that between C and G, sorry, C and T there, sorry. But we can say that between A and T there are 2 hydrogen bonds, and between C and G there are 3 hydrogen bonds. Lots of things we can say there for those 3 marks. Let's look at the mark scheme now and see what we could say. I'm kind of interested that this is even on the mark scheme here because it doesn't really tell us about the polynucleotide chains themselves. It says what holds them together. So I'm actually kind of surprised that that mark is even on there because it's not really talking about the bonding in between the chains. These are the marks that I would expect. So hydrogen bonds, purine to pyrimidine, or you could have given the letters A to T and C to G, and reference to the correct number of bonds. Okay, so that means A to T is 2 bonds, and C to G is 3 bonds, okay? Okay, on to question 23. All right, Sumatran rhinoceros. Can I just say, before we get started, the Sumatra rhinoceros is extremely, extremely rare. There are fewer than 100 of them. I think I looked it up actually the other day because I saw a news article about them. There's like 60 of them at the moment, which is so, so small, and they're only in one tiny little national park. So if you're feeling like you want to give to a good cause, look it up, give some money to protect these Sumatran rhinoceroses, or go visit them if you ever have the chance, and support some ecotourism because they're really in danger of dying out. Anyway, let's get on. Okay, so let's complete this thing here. So, well, let's do this one first. Okay, they're animals. Okay, so Animalia, kingdom, phylum, class, order, family, genus, species. There's so many different things you can remember this with. Like the traditional one is King Philip came. No, it's all right. Yeah, King Philip came over from Great Spain is the traditional one. But there's loads of others that you can look up. Some of which are like a bit more rude than others. So, or you want to make up your own one, that's also a good idea. Oh, I didn't do this one. Now, how do I know? I'm not supposed to know this. How am I supposed to know what species is this Sumatran rhinoceros? Well, it says it up here. Dicerohinus sumatrensis. And you need to remember that this here, this binomial naming system, is the last two bits. So I can't even say this. Dicerorhinus is the genus, and sumatrensis is the species. Okay, I'm going to pop in a little picture now of these lovely guys. There it is. That's gone. Okay. The name Dicerorhinus is part of the binomial system. State and advantage using this system rather than using common. Okay, well, this is basically like language issues. The fact that you can refer to it globally, different biologists can refer to it using this name, and we can be very specific. Let's just double check the mark scheme there. Dun, dun, dun. Class, order, genus, Animalia, sumatrensis. Oh, look at this, by the way, before we move on. Oh, did I get a mistake here as well? Sumatrensis must be all in lowercase. Oh no, did I get that wrong as well? Yes, I did. I said it in lowercase. Okay, good. Got to be in lowercase because for the binomial system, you have uppercase genus and all lowercase species, okay? So what I was talking about here, universal, recognized worldwide, accept no language barrier. And also it communicates, this is important as well, that it tells you the genus and species it belongs to. It gives you more information than just the common name, Sumatran rhino. Why is it critically endangered in the wild? Well, I'll tell you why. The main reason is habitat loss, so that's deforestation. Principally due to things like cutting down forests for palm oil plantations. So try to avoid using palm oil in all your products if possible, or buy Rainforest Alliance certified products that are sort of free from deforestation, or try to reduce the impacts of deforestation. So let's look at the mark scheme for this. So yeah, loss of habitat, that's the key thing. Deforestation, this is the same point, all this bit here. The next thing, hunting, poaching for horn, or climate change as well. Yeah, those are two other points. So notice that, I was being quite quick when I explained that. It's a 2 marker. So therefore, you've got to say 2 things. So number 1, deforestation, and then you're thinking, well, what else is going on in the world? Poaching, climate change are good answers. The remaining population of D. sumatrensis are all small and are scattered in isolated areas. These are factors that might speed up the extinction of the species. Well, when the populations are scattered in isolated areas, that means they can't get together to breed. So breeding becomes more difficult. And we can also get, if you have multiple small populations, those small populations can actually become sort of inbred, which is something called, it leads to a loss of genetic diversity within the population, which is something called genetic erosion. Let's look at the mark scheme here. So it's hard to find a mate, maybe a gender imbalance, inbreeding leading to low genetic diversity, This is actually called genetic erosion, small gene pool genetic bottleneck. And because they're dispersed, they can be less likely to adapt if they have low genetic diversity. So point 3 kind of follows on from point 2. Yeah, these, 1, 2, and 3 are for me most relevant. 4, 5, 6, yeah, they give them to you. I slightly disagree with them because actually, if the populations are multiple small populations, they're less likely to all be wiped out by the same disease than if it's one population together. In actual fact, this population is all in one national park. So I even read in this article the other day that if there's a massive tsunami in Indonesia, which there are from time to time, it could wipe out the entire population in just one go. So we really hope that we can maybe reintroduce them to other areas to allow them for their population to kind of re-establish. And that's kind of what the next part of this question is about. Captive breeding programs have been unsuccessful, suggest another way in which zoos can contribute to the conservation of the Sumatran rhinoceros. Well, things like education, things like increasing the area of land for the national park, that sort of stuff. Those are things that you could do in the MARC scheme. Education awareness, support for promote conservation projects or research. And there's things that are listed here. Raise money, provide funds, technical support, sending people to monopopulations in the wild, supporting the setting up of a nature reserve, and that sort of thing. So, so I mean, for that one, there's a lot of kind of examples that you could have said. So if you think your example fits in those 2 points, then go ahead and give yourself the mark. Okay, moving on to 24. All right, okay, so in mammalian blood, oxygen transport with hemoglobin, the presence of hemoglobin greatly increases the oxygen carrying capacity of the blood. So we've got some figures here. 100 centimeters cubed of plasma contains 0.3 centimeters cubed of oxygen when fully saturated. Plasma is the liquid without the red blood cells, okay? 100 centimeters blood contains 20.1 centimeters of oxygen when fully saturated, okay? Big difference there. Calculate the percentage increase in oxygen carried in fully saturated blood compared with oxygen carried in fully saturated plasma. So we're looking, so remember, percentage increase is about, is equal to the change, got to remember this is very, very important, divided by the sort of starting value times 100 to make it percent, okay? So what's the change? The change is 20.1 minus the starting value of 0.3. And then the starting value is 0.3. And we've got to times that by 100. So if we do a little bit of working, the difference on top is 19.8 divided by 0.3. Then we times it by 100. So 19.8 divided by 0.3 equals, times it by 100. Yes, it is 6,600%. Okay, I can see that you might have maybe left it at 66%, but I think 66% would be less than doubling increase. So if you double something, that's an 100% increase. But this is a much bigger increase than that. So yeah, it is, 6,600% increase. Massive. There it is. Okay. Hemoglobin is found in erythrocytes. Unlike other vertebrates, the mature erythrocytes of mammals lack nuclei and other membrane organelles. Explain one advantage and one disadvantage of the lack of nuclei and other membrane-bound organelles to mammalian erythrocytes. Well, the main thing, the advantage, is there's more space for hemoglobin. Disadvantage is they can't replicate, can't produce new proteins, that sort of thing. I actually wasn't aware that it was so rare, and it was, okay, it's mammals, isn't it? So I think birds do have nuclei in their red blood cells, but mammals don't. I didn't realise that it was only mammals that didn't. Let's look at the MARC scheme, just double check. So here we are, advantages, the one that I said, more space for more hemoglobin. This is another good point, which I hadn't thought of. Can squeeze through capillaries more easily. Disadvantages: limited lifespan, cannot divide, cannot reproduce, or cannot undergo mitosis. Also, no protein synthesis or repair. Yeah, this is also true. There's no respiration in mitochondria. But if you wanted to talk about that, you couldn't just say no respiration, actually, because there is some respiration, which is anaerobic. You'll learn more about this in year 13. Aerobic respiration requires mitochondria, but anaerobic respiration does not. Okay? All right, moving on. Viruses do not use erythrocytes as host cells, whereas the malarial pathogen Plasmodium spends part of its life cycle inside erythrocytes. Okay, well, let's kind of think about this. Well, what do viruses need? Well, viruses need machinery to copy themselves. They need things. They need things like DNA polymerase, RNA polymerase. They need ribosomes. They need, they need enzymes. So DNA pol, RNA pol. In order to replicate, they need lots of ribosomes to replicate and so on, because they basically just take over the mechanisms of the cell. Whereas the malarial pathogen, essentially what it does is it pretty much just eats hemoglobin. Okay, it has its own, it's a cell. Okay, it has its own machinery to grow and to replicate itself. Okay, so that's the real significant difference. In the mark scheme here, that's what we find. So virus cannot replicate itself, so it requires, this is what I was getting at, I think I would have got the mark, requires host cell machinery. So for example, ribosomes. Viruses does not contain its own ribosomes, it needs ribosomes, whereas the Plasmodium is using the host cell to hide from the immune system. I didn't really say that. I did say this, okay, so Plasmodium to use as a source of food. Plasmodium to use as a source of food for growth reproduction. It's kind of just eating the hemoglobin, but it is its own cell, so it has its own machinery to copy itself, okay? Ignore it has its own DNA nucleus protein. Yeah. Yeah, okay, so those are the marks. Moving on. 2. Explain why erythrocytes do not make use of any of the oxygen that they are transporting. Well, this is because they have no mitochondria, therefore no aerobic respiration. Like we discussed earlier. Mark scheme, check. Yeah, lack mitochondria, therefore no aerobic respiration. Also, other things you could have said, oxygen is firmly bound to hemoglobin while being transported. And also, if you're thinking about, well, why don't the red blood cells need to do any respiration? Well, they don't really need much energy. They just get carried along. They get moved by the mass flow of the blood. So they don't need any ATP or energy to move, or really for any metabolic processes, because there's no transcription or translation of proteins going on because they don't have a nucleus. Okay, on to this one. Oxygenated blood returns from the lungs to the heart before being pumped around the body. So this is going to be a cardiac output calculation. Normal cardiac output is 5 dm³ per minute. 100 cm³ of blood contains 20.1 centimeters of oxygen when fully saturated, and that's nicer than they put that there. It was already up there, but they've written it down here. Calculate the volume of oxygen transported to the tissues per minute to 4 significant figures. Okay, right, bit of maths now. Right, first thing, what's a dm³? 1 dm³ equals 1000 cm³. Okay, so let's take this figure and convert it up, okay? 100 centimeters of blood contains 20.1 of oxygen when fully saturated. Well, what would 1000 cm³ of blood contain? So 1000 cm³ would contain 201 cm³. But 5 dm³ per minute is 5,000 centimeters cubed per minute. So basically 5 times 201 equals what's that? Sorry, calculator, come on. 5 times 201, 1005. Okay, 1005 centimeters cubed, which is correct. With reference to the structure of blood vessels, explain why oxygen is not released until the blood reaches the capillaries. With reference to the structure of the blood vessels. Okay, so I think we're talking about, so now, to be honest with you, I haven't prepared this before I'm recording this, so I'm sort of a bit stumped here. So what do you do when you get a bit stumped? Well, okay, so before you get to the capillaries, you've got the arteries, and then you get the capillaries. We need to talk about structure. Okay, so I'm not really sure if this is going to work, but let's give it a go. So arteries, you know, it's sort of tough, elastic, and there's no tissue fluid formation. I think that might be important. Capillaries, narrow, leaky, holes, tissue fluid. And maybe we can also say that CO2 from surrounding tissues gives the Bohr effect, which causes dissociation. Although that's not really about the structure of blood vessels. So, you know, that would be my guess. You know, sometimes you do have to kind of just think, what is the question really wanting from me? Let's see what the mark scheme says. Let's see if I got it along the right lines. Okay, so yeah, arteries and arterioles have a thick wall, whereas the capillary wall is only 1 cell thick. It's about diffusion. Of course it is. Okay, so I didn't quite get there. I might have got 1 point there out of 2. Disappointing. Yeah, so the capillary wall is only 1 cell thick. You can't have diffusion through the artery wall. It's too big, or the diffusion distance is too large. So really the key thing, which I sort of forgot to mention, is diffusion. Oh, notice the Bohr effect is coming up for the next question, which we already started to think about. What is the next question? Well, here, classic. Okay, so D. Figure 24 shows the oxygen dissociation curves at different carbon dioxide concentrations. The name of this shift, so when we increase CO2, hemoglobin sort of releases more of its oxygen. This is called the Bohr shift or Bohr effect. Letter T in figure 24 indicates the partial pressure of oxygen in actively respiring tissues. Explain why the blood offloads more oxygen to actively respiring tissues than to resting tissues. Okay, well, what we can do is we need to get some figures out of here. So we want to probably kind of go there and look and see the percentage saturation. So that's 24. So 24, the hemoglobin will be 24% saturated in actively respiring tissues if there's high CO2. But without high CO2, we get 40% saturation. So AR actively respiring tissues, ART actively respiring tissues, I'm just writing shorthand, is going to produce CO2. Percentage saturation drops from 40% saturation to 24% saturation, sort of offering or unloading an extra 16% of that sort of bound hemoglobin to the tissues. Let's have a look here. So yeah, high CO2. Yeah, and then we get this, I got this mark here, so drops from 40% to 24% saturation, or a 16% reduction. Stuff which I didn't say, this is a 2 marker, but it's always good to try and get more than the max marks. Does that sound stupid? Maybe it does. If you're going for a 2 marker, say 3 things, because then even if you get 1 you miss a keyword on one of the points, you're going to get the other point. You're still going to get 4 marks. Always go for more marks than are available. So the other things I should have said, lowered affinity of hemoglobin, and actually the mechanism for that, which we didn't really go into. I didn't think the question was looking for it, but the mechanism of it is the carbonic acid dissociates and it causes an increase in H+. This binds to the hemoglobin and slightly alters its three-dimensional structure, its quaternary structure, which releases some oxygen. Okay, so that is 24 complete. Okay, we're making good progress. We're going to move on to 25 now. 25. All right, the following statements. Summarize the results from experiment design. Discover more about the translocation of organic materials in the phloem. State all letters that provide evidence for the following conclusions. Okay, right. Okay, active and source to sink. Right, so what I'm going to do is I'll do translocation as an active process, and then I'll do sugars are translocated from source to sink. And let's look through. Any increase in the sugar content of leaves is followed by a similar change in the sieve tube contents in the stem. I would say that's the source to sink one, because it shows that leaves then follow into the sieve tube. The rate of transport increases the nutrient. Okay, so this is to do with active processes, because above 25 we've got maybe some enzymes denaturing. Enzymes, they're active. Translocation stops when stems are treated with substance that inhibits respiration. Okay, well that's definitely to do with active respiration is needed. D, sugars can be transported both up and down the plant. Okay, well that's source to sink, because sometimes something like a potato can be a source, but like in the winter, okay, when the plant is not actively photosynthesizing as much, stored starch in the potato might get converted back into sucrose and then moved up to the rest of the plant to keep it going. Aphids can be used to sample phloem sap. So what? I don't even know if that provides any. I don't even know if that's anything. I'm going to leave that one. We'll see if I'm right in a minute. Roots, young leaves, and growing fruits will import sugars. Okay, that's source to sink. So I went for B and C, and then here A, D, and F. Tension, am I right? Yeah, okay. So B and C and A, D, and F. Okay, so the random thing about aphids caused me a bit of anxiety there, because I was like, oh, well, I don't know where to put this one. But yeah, that was a trick question. Aphids, that thing doesn't really tell you anything. So you just have to stick with the courage of your convictions and just leave that one out. Right, explain how mass flow of the phloem sap occurs in plants with a vascular system. Ooh, last one. I didn't realize we were at the end of this paper. I thought we had more to go. Okay, so, well, we've got, here's the phloem. We've got a companion cell, with its nucleus and all that stuff and, stuff. Okay, so what happens? The companion cell, first of all, it actively pumps out H+. I think that's right. Then the H+ goes back in with glucose. Okay, this is called co-transport. So glucose is carried back into the companion cells. Now that glucose diffuses through the plasmodesmata into here. So we have loading, okay, active loading of glucose here. What happens when you put lots of glucose in a place? Well, water follows via osmosis. So I'm going to do this light blue here. Water follows, and this generates a bit of a pressure. Then we get mass flow. Mass flow occurs down to the sink. Let's change color. Let's do black for the sink. And in the sink, we can have unloading of glucose, which means that water exits as well. So let's look at the mark scheme. Okay, sugar, sucrose, or assimilates in the sieve tube. They enter the sieve tube, which lowers the water potential. Notice we have to talk about water potential. We need that and there. It enters the sieve tube and it lowers the water potential. It moves down a water potential gradient. And oh, look, okay, I'm rushing because it's the last question in the paper, but I'm missing so many marks. Hydrostatic pressure. I just said pressure. Okay, look at these underlined words. They're so important. Increases hydrostatic, which means the physical pressure. And then at the sink, we have the glucose, or sorry, the assimilates or the sucrose leaving the sieve tube, active removal, which and when we remove, that increases the water potential inside the tube, which causes water to leave the tube via osmosis down the water potential gradient. Okay, this lowers the hydrostatic pressure. We've really got to talk about this pressure, which means that we get this mass flow. Although, yeah, it's not given the mark, is it? Because we don't get the mark for mass flow because it's already given in the question. It's saying explain why mass flow works. So we need to really, I botched this question. We need to say that the flow is from a high to a low hydrostatic pressure, okay? Just shows that when you're rushing and you think, oh yeah, I know this, I'm eager to be finished with this paper, pause, think about the vocabulary you will use, because even, you know, biology teachers like me, I've been doing this for like, what, 15 years, can forget to say the keywords if you get a bit hasty, okay? So think calmly, think slowly, think keywords. All right, that is everything. I hope you found that useful. If you didn't catch something I said, obviously you can rewatch a certain question. You could even try the paper again in a week's time after this video has slightly faded from your memory and see how much you recall. Okay. All right, and I'll see you on the next run through video. Thanks.